Okay, so now let's see how the virus infect more closely. We just mentioned that the virus binds to a receptor on the lung epithelial cells. Actually, it is the virus surface protein, which is called spike protein or S protein, that binds to a receptor called ACE2 on our lung epithelium. So what kind of antibodies may block this process? Yes, most of the time it is the antibodies that binds to the spike protein, or in particular, the receptor binding domain, or RBD, on this spike protein that can block its interaction with the ACE2 and then block virus infection. Okay, so how do we get those neutralizing antibodies? There are actually three ways to develop an antibody drug. First is, of course, we can just purify it from a patient. We can get the B cells from the patient and find one clone of B cells that express a very good neutralizing antibody, and then we use this to produce large amount of neutralizing antibodies. And these are called patient-derived neutralizing antibody. And also we can just use a lab animal like a mouse. We immunize this mouse with vaccine, with the virus or deactivated virus, or even we can just immunize them with the S protein. And then this mouse will produce antibodies against this virus. And it's similarly, we can just get the B cells and then produce the antibody. The third strategy is that we don't use any kind of animal at all. We just express a fragment of the antibodies on a small bacterial virus called phage. We can express a lot of different kinds of antibodies. We call a library of antibodies on phages. And then we use the S protein from the virus to fish out the one that combines to it. This is another way that's called phage display. So with these two ways, we don't have to depend on the patient to get antibodies from for this virus. Okay. Now again, does this work, right? Do the neutralizing antibodies work? Well, there are different stages of testing whether it works or not. For example, we can test it in cell culture to see whether this antibody blocks the virus from infecting the cells. Or we can test it in an animal model. And eventually, we have to test it in human to see whether it's really working. Okay, but currently, again, it's still too early to say. The published results are mostly on this cell culture. So people have identified a lot of neutralizing antibodies from the patient or from phage display or from previously infected like SARS animals that they can all neutralize these antibodies from infecting the cells. But actually, a drug development process is quite a long process. See, most of the time, it takes years to develop a new drug from scratch. For example, we can see these two diseases that we heard a lot in the recent time, Ebola virus and MERS. We can see that the Ebola virus outbreak is in 2014 and MERS in 2012, but currently we still have no available neutralizing antibodies in the market to treat these two diseases. But now, hospitals, companies, and academia are working together to put into a lot of efforts to accelerating this drug discovery process for COVID-19. We can see in these slides, there are a lot of companies working on this. And the estimated clinical trials may start as early as this summer. And yeah, so this is the case of neutralizing antibodies. Okay, now we talked about the neutralizing antibodies that bind to the spike protein. So does it have to be an antibody? Does anything else bind to the spike protein? The answer is yes, right? We all know that this ACE2 receptor binds to the spike protein. So we can also use a fragment of the ACE2 receptor to compete with the S protein. These are called binding competitors. So here there are also some studies showing that ACE2 receptor and other drugs that derived from it can block the virus infection. And also, when the virus infects the cells, the S protein will undergo a conformational change to infect the cells. So people have also developed fusion inhibitors, which is a piece of peptide from the S protein itself that can block the fusion, this conformational change, so that the virus cannot infect the cells. So these are the alternative approaches that target this virus infection. All right. And then, so we just target this S protein of this virus. Now I'm having another question. So there are two binding partners, the S protein and the ACE2 receptor. Can we target the ACE2 receptor to compete with the virus binding? These kind of agents can be an antibody or an RBD mimetic, right? So actually there are publications on these. For example, this is SARS-CoV-2 RBD, which can inhibit the virus infection. However, this strategy has some concerns because the anti-ACE therapeutics are shown to may interfere with this ACE biological function, or it may lead to upregulation of ACE2, which may not be a good thing. So when we're developing this kind of therapeutics, we have to keep in mind of these concerns. All right. Now I'm gonna to summarize this part. To block the virus infection, we can either target the spike protein or the ACE2 receptor, and we have a lot of different ways, like using antibodies or using mimetic of the protein to block this process.